It's my pleasure to introduce Dr. Jay Shendure. Jay is a professor in the Department of Genome Sciences at the University of Washington, an investigator at the Howard Hughes, and the scientific director of Broadman Beatty Institute for Precision Medicine. He also leads the Seattle Hub for Synthetic Biology, which is a collaboration between the Allen Institute, CZI, and the University of Washington. Jay has been at the forefront of DNA sequencing technology for nearly 2 decades, from his early contributions to next-gen sequencing to pioneering work in massively parallel functional genomics, single-cell genomics, cell lineage tracing, and embryo-scale developmental atlases. His contributions have been recognized by elections to the National Academy of Sciences and Medicine, and also the NIH Director's Pioneer Award, among many, many other honors. Please join me in welcoming Dr. Jay Shendure.
Thanks, Yash, and thanks, Jeremy and others for the opportunity to be here. Okay, so I'm going to talk about a specific topic here, recording and barcoding of monoclonal models of early development. Maybe just to dive in, I'll kind of set this up in terms of the motivation. So, when we think about cell lineages, I think the first thing that comes to mind is the, seminal lineage of C. elegans, which was deciphered by John Sulston and colleagues some 40 odd years ago. I think one point that's important to recognize is that invariant lineages, such as that we see with C. elegans, where it essentially happens exactly the same way every time, which is partly what enabled him to get the lineage in the first place, are the exception and not the rule, right? This is only a very small number of species, animals, that have this kind of invariant development. The rule is regulative development, right? So across animals and plants, lineages are inconsistent and yet somehow give rise to consistent form and function. And so you can think about the genome as a blueprint for that process of the variable processes that give rise to a consistent outcome. And this is what I would call mysteriously precise, right? You think about twins. We know their lineages, the cell lineages that give rise to identical twins vary vastly, but somehow the constant genome programs a consistent outcome. And even across, mouse versus human, say, if we look at phenotypes, such as the c-KIT mutation, piebald phenotype shown here, we see consistent features that derive presumably from shared patterns across, between the lineages that are disrupted in consistent ways, leading to the patterns seen there that are common between mouse and human. But how this unfolds in detail, it remains a mystery. Okay, so endless forms most beautiful. That's a phrase from Darwin talking about natural species and what we see that evolution has created. The modern equivalent of this is in a dish, right, a cell culture hood, where with organoids we can increasingly create a diversity of form and functions and model, that occur in a human or a mouse. But we can model these in a dish with embryonic stem cells that are differentiated under various protocols, right? But a challenge in this field, right, is that in contrast with the precision that we see in nature, there's quite a bit of variability encountered in the dish, right? So here, this is just kind of an exemplar from this image from Stefano Vianello showing different gastruloids, generated through a protocol and just the quite a bit of variation in form and the extent of different cell lineages that are represented, right? So this irreproducibility is on one hand quite annoying if you're trying to study phenomena, but it also is quite interesting, and I think gets to the some key questions, around what bounds regulative development, what makes some protocols more precise than others, and how might this variability be exploited in actual in vivo development, right? Just for context on gastruloids, so these are a powerful system developed some years ago by I think now over a decade, to model early development. And essentially this is modeling the early posterior embryo, whether it's a human gastruloid or a mouse gastruloid, It's a quite straightforward protocol where you apply a Wnt agonist to an aggregated collection of embryonic stem cells, and it exhibits, some of the classic features of gas relations, such as symmetry breaking. By and large, I think these are well characterized at this point, and we understand that, we see differentiation to neuromesodermal progenitors, which are precursors for the lineages that give rise to kind of the bottom half, right? The spine, including posterior neural cells of the spinal cord and paraxial mesoderm of somites, right? And so, however, many implementations of this protocol give rise to inter-individual variation. So this is data on the bottom from Prisca Liberali's lab and on the right from John Marioni's lab, essentially showing that if you look at individual gastruloids, whether through the lens of single-cell RNA-seq or imaging, where certain lineages have certain markers, you see quite a bit of inter-individual variation, right? So every bar there in the bar plot is an individual gastruloid. You see differences in cell type composition. And then along the bottom, we have individual gastruloids, which vary as well. Okay, so changing gears a little bit and intersecting this with technologies that we in the broader field have sought to develop around molecular recording. DNA Typewriter is a method developed by Jinhong Choi, who was in my group at the time. He now has his own lab at Memorial Sloan Kettering. And in a nutshell, this is a way of symbolic recording to DNA. So kind of taking methods like Gestalt, which we and Alex Shear's group developed on the order of a decade ago now, and trying to take them to another level by introducing the concept of order, right? So we're writing events along DNA from kind of left to right, 5 prime to 3 prime, and also symbols. So we're actually, putting in insertions that encode more information than a simple stochastic indel, as you might occur with the double-stranded break, right? And so how this works in practice involves prime editing and what we call a DNA tape, which consists of a set of repeating 14 base pair monomers. And the monomers are designed such that the initiating monomer with the red box there on the top left, together with the first half of the next monomer, and this preceding key sequence, shown there by the 3 red bases, GGA, comprise a target site for a prime editing guide RNA. And the nature of the edit encoded is going to be an insertion. And the insertion accomplishes 2 things. One, it introduces a barcode, which encodes some piece of information that we're interested in recording. And also it inserts a key in front of the next monomer in the tape, right? So in one step, we've recorded information, we've inactivated one of the monomers, and we've activated the next monomer, right? And so I'm going to skip over, this is published some years ago now, so showing why and how we believe this worked. But when we integrate this with single cell profiling, we get images or data structures like this. So every row here corresponds to a single cell, here profiled by 10x. And we have 11 of these 5-unit barcodes. And the colors correspond to different stochastic insertions, 3 base pair insertions plus the 3 base pair key for a 6 base pair insertion. And you can see within any given unit, we're writing left to right. And for this particular set of 24 cells, you can see that there's information about lineage encoded in kind of the stochastic insertions that allows us to cleanly separate these 2 clades as well as discriminate some of the subclades. And so now this scales with our single cell technologies, and we can generate lineage trees much larger than what we see with from the Selston tree through this kind of retrospective reconstruction. And this also cleans up a lot of other issues with Gestalt and related technologies. So far, up to the point of the proof of concept, we had done this in HEC293 T cells. We've been since kind of pushing these towards the gastruloids that I mentioned earlier, and trying to answer questions around this observed heterogeneity that I mentioned. And so, if you think about how one makes a gastruloid, or really most organoids, you're not starting with a single cell. You're starting with an aggregate, a polyclonal aggregate of many cells that you then differentiate in some way, right? And this is true of gastruloids as well. And so one concern before we even started down this path was whether some of the heterogeneity that you see within a batch of gastruloids performed with a single protocol within a single lab might be due to heterogeneity amongst the aggregates that you're actually subjecting to this protocol. So if this collection of cells happens to differ between the first and the second aggregate, you might see different outcomes, right? So towards controlling this, but also for some other reasons, Sam Regalado, together with Troy and working with Chengshang Q, who now has his own lab at Dartmouth, worked on trying to essentially come up with a monoclonal protocol for making gastruloids, right? Essentially we start with now a single cell, and we grow that up and make a monoclonal aggregate, and then subject this to the conventional gastruloid protocol, right? So here, we get things that look and feel like gastruloids. This is a longer protocol because we're growing this single cell up into a monoclonal aggregate. But in a sense, we've eliminated some of the heterogeneity, hopefully, by guaranteeing that the cells in this aggregate are closely related to one another, or at least more closely related to one another than they were with the polyclonal aggregate. Right? So that's been one sort of big change. To our surprise, right, when we do this, we can, of course, barcode the cells. So introducing static barcodes that essentially identify every starting cell. We grow up one gastruloid per well. We then sort of we essentially take all of these, we profile them in a pooled format with single cell RNA-seq. We also perform debris-seq, essentially sequencing material that's left over in each well. And what the combination of these things allows us to do, together with these that we know that these barcodes uniquely define each founder cell, is to take this 10x data and disaggregate which cells came from which wells, right? And so we get these nice assignments based on debris-seq and single-cell RNA-seq, so we can group the cells based on the wells that they came from. The product of this, it wasn't to actually decrease heterogeneity, we actually ended up increasing it, right? And so if we look at these monoclonal gastruloids, we now go from what before we would largely see two categories. We would get gastruloids, they would tend to be either biased towards mesodermal lineages or biased towards spinal cord-like neural lineages. But now we get, in more abundance, these two additional categories, one of which I'll call on the left here kind of a failure to launch. These things stay very ESC-like. And on the right, they're neural in terms of the cell type compositions, but these look more like the hindbrain than the spinal cord cells that originate from neuromesodermal progenitors and are more typically seen in gastruloids, right? So now we have these four groups to work with. And so, convenient, retrospective explanation here, which I think is valid in light of some of our later data, is that the polyclonal nature of the aggregates was serving a role to essentially buffer some of the heterogeneity that may be present at the single cell level. And now when we make the founder a single cell, we end up unmasking some of that heterogeneity by eliminating that source of buffering. So now we reframe the question a little bit because to kind of think about when is the origin of intra-batch heterogeneity, right? So rather than where, when along this path. And so this is where we turn to the typewriter technology. And so here now we have, imagine a stem cell, a mouse stem cell, in which we have many of these tapes integrated throughout the genome. We also have many pegRNAs that are integrated throughout the genome, driving different insertions to the tape. And we have an inducible version of PEMAX, which is a newer prime editor. So like what I showed with the HEC293T tree earlier, we start with a clonal founder. We then might actually make a bunch of founder cells from that clonal founder and use each of them together with induction of docs here to start the typewriter to make a monoclonal gastruloid, right? And then we perform kind of the RNA-seq, tape-seq, and debris-seq. So the resulting phylogenies look like this. So this is two gastruloids that originated from the same founder cell. We can see that, and we see the same sort of information now presented a little more colorfully. We now have the added information of cell type, which is more variable than with HEC293 T cells. But again, we can reconstruct this nice lineage. And the lineage information nicely correlates with our debris-seq information in separating this into two subtrees, which correspond to the individual gastruloids. Right, so here showing this now in a circular form. We've got the two gastruloids. We can see that especially for the older branches or the older parts of the tree, our bootstrap values are quite good. And so we're confident in that structure. And you can see along the outside, we're presenting the cell type labels. And there's a lot of intermixing, but there's also some structure, right? Which is interesting, right? And so as we kind of break this down in more detail, we see some really interesting patterns, right? So here, so this is now, again, I'm showing the tree, lineage trees in a circular form. Now we're only looking at a single gastruloid, and we're focusing on a single cell division, an inferred cell division. And the numbers here correspond to bootstrap values. And I'm telling you what cell division it is relative to the founder of this particular gastruloid. And here you can see at this cell division, I would call this a fate restriction, where all of the descendants, 100% of this, this daughter over here, were extra-embryonic endoderm, right? Whereas we see a mix of outcomes in the daughters of this other, daughter of this particular branch point, right? And so this is a fate restriction where this bottom lineage in particular was restricted extra-embryonic endoderm. If we look at another lineage, we see, or another example, this is a third cell division of a different gastruloid. We see a pattern where, again, very high bootstrap values, but now we're splitting between a mesodermal bias lineage versus a neurally biased lineage. I would call this, this is more like fate bias, not fate restriction, because we still see we see a diversity of outcomes, but they're clearly biased between this set versus this set. This is another really fun example where now it's the first cell division of a gastruloid, and we see that one of the daughters exhibits this, the vast majority of its daughters and granddaughters, etc., exhibit this failure to launch, right? Whereas the other daughter has descendants that look more like what we would see in a conventional gastruloid, right? Again, fate bias, though, not fate restriction. And so the really cool thing here is that these early cell divisions are before we apply any chiron or any sort of Wnt agonist to actually make the gastruloid or the aggregate go one way or another, right? So these cell divisions are when this monoclonal founder is still growing up into an aggregate, right? And so we can't, like one hypothesis is that these are cell intrinsic fluctuations in stem cells that are heritable. I mean, they're stochastic, but they're also heritable. They must be heritable because they are persisting through to when we actually, have an aggregate and perform induction of the gastruloid. But there are also other explanations. Lineage is correlated with spatial substructure, and so it's entirely possible that there are spatial correlates of lineage that are playing out here, and so certain cells may be biased or lineage is biased towards the interior of the gastruloid versus the exterior. And there's other threads of work from folks like Jared Tochter that support that this is maybe part of the story as well. I only have 2 minutes, so I apologize that I'm speeding through this part, but I think it's pretty cool. So towards answering this question, we perform what I'll call a founder of founder experiments, right? Where now, rather than activating the DNA typewriter here, we're activating it way back here with, in the first, when we're first growing up the clone that is then going to give rise to the stem cells that will found the individual gastruloids, right? And so we essentially get a tree of trees where, we've got a founder cell, gives rise to many descendant founder cells, each of which gives rise to a gastruloid. And we can ask whether gastruloids that are more closely related give rise to, founders that are more closely related give rise to gastruloids with similar cell type compositions. And in a nutshell, the answer is yes, they do, right? And so now that, we disaggregated the cells prior to making, founding the next generation of gastruloids, so we've eliminated the spatial component of information, but nonetheless, we see that this effect persists, right? The gastruloids are now much more similar to one another, which might be due because the founders are more similar to one another. But even within that set, we see that more closely related founders end up giving rise to gastruloids that are more similar. So we've subsequently done another experiment along these lines. This is like a a Samantha Morris or Arjun Raj style, like rewind type experiment, where we're now trying to gain information about the transcriptional and epigenetic states of the founders and relate that to the outcomes of the gastruloids that ultimately emerge. And the analyses of these data are still in progress, but just to kind of cut to the chase here, we do in fact see that the epigenetic states of the clonal founders um, are informative with respect to the predictor, what the outcomes, right? So I'm cutting through a lot here, but just to get to the heart of it, if you look at embryonic stem cells, not without, just a bunch of them, right? And you do ATAC-seq, let's say, you get a big gamish. This is a pretty, there's heterogeneity, but it doesn't really have a lot of structure that's obvious. If we then make gastruloids from these, we see that, as we saw before, that the monoclonal gastruloids are biased towards different outcomes. Some are epiblast bias, some are hindbrain bias, some are somite bias. And now if we look backwards, specifically at the clones that gave rise to these kinds of biases downstream and analyze that, with that lens on, we do in fact see that, differences and subtle differences in their epigenetic states that correlate with the biases that are going to come downstream in terms of which TF motifs appear to be active in those founding stem cells or their sisters, so to speak. So we're very excited about that, and we think this fits with other bits of the literature in an interesting way. So we're not saying space is not involved, it certainly is, but this is, this other aspect is also relevant. I'm going to skip over the rest for reasons of time. We've gotten to big trees now, 50,000 cells in a single monoclonal tree where we have cell types at the leaves, all descended from a single founder, and overall comprising, 96 gastruloids that are, share common descent. And we think these technologies are primed for further scaling with, as single cell technologies get cheaper and recording methods get even better. So thanks for your attention, and look forward to any discussion.
So what is the path to applying DNA typewriter to the mouse embryo itself rather than an organoid model? How do you see that becoming possible?
Yeah, I don't think it's that. I don't think we're that far from that. I guess the challenge, like there's the quick and dirty, and then there's kind of the stable community resource type thing, right? And I think there's a couple of important things to note there, right? One is, provided prime editing works in vivo, and we know it does from work from David Liu and Jonathan Weissman and Tyler Jackson and others, and provided that one can recover tapes and all of that, it seems like it's within reach, right? And then we're limited by the scalability of single cell technologies, et cetera, right? And so I think the path is there, right? I do think there's a few, like I think looking, if you look further down the road, I think there's a few challenges, right? I think one, lineage trees in themselves are hard to make sense of, right? Especially when you're only sampling a small percentage of the cells. There's 500 million cells in a newborn embryo. So unless we're profiling half or more of them, we're not going to get the last few cell divisions, right? And so, and trying to work towards decorated trees with methods like engram and other kinds of signaling recording, I think are going to be important in the long run. And then I think trying to get, like George Church and others, and, when people do these things in vitro or even in vivo, often the components are quite scattered throughout the genome, which is, good, but doesn't make for kind of a single locus kind of thing, like what has been done with Fernando Camargo's Darwin mouse and things like that. So I think the devil's in the details, but I do think that the road is pretty clear now, if that makes sense.
How far do you think we can go to record in vivo mammalian development, let's say from a single cell stage? Do you think there's limitation on tape recorders and things?
I think there are technical challenges that are in like that'll that'll manifest is along that path, and we're running into some of those now. Um, but I don't think there's any barrier at this point to high resolution trees. It's just a matter of execution, right? I do think that um, I don't think it's going to take the form of a Gestalt type recorder, right? I think it's going to fall into a something that's prime editing or base editing based, right? I think there are advantages to those approaches. Uh, but I think the path is increasingly clear.
Do we know what these states are? Is it epigenetic within stem cells, or is it metabolic state? Is it
I
something
don't
to
I don't
do?
think we know. Yeah, I'm not sure we know, right? I think
Okay.
we're seeing these subtle differences in the epigenome, right? They're easier to find in the epigenome than the transcriptome, which is interesting, right? Because if you can aggregate signals across a lot of sites, that might end up actually being a more sensitive measurement of state than a sparse transcriptome, right? So that's pretty interesting. But that could also be reflecting, it's kind of like they're all just somewhere on Waddington's landscape sitting around there in the bowl waiting to roll down, but they're not at exactly the same position, right? But whether it reflects metabolic states and things like that, I'm not sure.